Hello, I'm David Attenborough, and today I'll be talking to you about antibody effector functions. At the core of antibody effector functions are B and T cells. Both are small cells with condensed chromatin and a small cytoplasm. The B cell expresses antigen-specific B cell receptors, also known as immunoglobulin membranes. When the B cell receptor is activated, the B cell becomes a large plasma cell which secretes antibodies. The antibody then binds to specific epitopes on an antigen. Unlike B cells, T cells do not recognize antigens directly, but rather fragments displayed on the surfaces of the other cells by special major histocompatibility complex molecules. An antigen is a molecule capable of inducing an immune response on the part of the host organism, though some antigens can be part of the host itself. Antigens can be classified according to their source: within the body, outside the body, or within proteins and protein complexes. Immunoglobulin plays a key role in the war between antibodies and antigens. Antigen binding site is located in the variable region on the light chain. The variable region allows each antibody to bind to a specific antigen. The constant region on the antibody does not vary the same way. Both chains together contribute to the antigen binding site. There are several processes involved in the detection and destruction of an antigen. First, there is antibody-dependent cell-mediated cytotoxicity, or ADCC for short, which is triggered by interactions of target-bound antibodies that contain Fc receptors known as FcRs. The process of ADCC includes the killing of antibody-coated target cells by the release of cytotoxic granules or expression of cell death-inducing molecules. ADCC is used for large parasites that cannot be phagocytosed by a phagocyte. Antibodies are produced to coat the surface of the particular parasite, and phagocytes are attracted to the antibodies. The defense cells then release lytic enzymes, which act to destroy the cell by apoptosis. Then there's the complement system, which is a system of inactive proteins in the blood that become active when they detect a pathogen. The system works in three ways, one of which is the classical pathway. The classical pathway is triggered by the formation of the antibody-antigen complex. The Fc region is at the tail region of an antibody, which reacts with cell surface receptors. The Fc portion directs the biological activity of the antibody. The Fab portion of the antibody has specificity for binding the epitope of the antigen to the antibody. The classical pathway of activation forms C3 convertase, which splits into two fragments: C3a, which is used to activate mast cells, and C3b, which covalently attaches to the surface of microbial pathogens and opsonizes them. The activation of mast cells causes the release of vasoactive mediators, such as histamine. The vasoactive mediators result in actions such as inflammation, smooth muscle contraction, pain, and chemotaxis. Still with me? Good. Just a little bit further now. Opsonization is the immune process of coating a particle with proteins to target it for destruction by phagocytosis. Proteins that carry out this process are called opsonins. An opsonin is an antibody which binds to foreign particles or cells in the body in order to make them more susceptible to phagocytosis. Immunoglobulins, complement proteins, and circulation proteins are all examples of opsonins. Opsonization therefore greatly increases the ability of phagocytic cells to identify foreign antigens, so that phagocytosis can occur, destroying the foreign particle. Next up is neutralization. Neutralization occurs after target pathogens have been recognized. Neutralizing antibodies, or NAbs, are involved in neutralization. The main differences between neutralization and opsonization is that neutralization involves disabling a pathogen's ability to bind to a target cell. This is also different to ADCC, which kills already infected cells to prevent the spread of infection. Agglutination is the last antibody effector function we will talk about today. This is the visible expression of the aggregation of antigens and antibodies. The process results in the formation of an antigen-antibody complex, which expresses itself in the form of a clumpy substance. Thank you for listening, and have a pleasant day.